Hello everyone, welcome to GTech Techno Solution Private Limited. Today we are going to discuss about the glomerular filtration rate, which is abbreviated as GFR, and we are also going to study about the DCT PCT Tubules, which is nothing but your distal convoluted tubule and proximal convoluted tubule that you can find in your nephrons. So these are the topics that we are going to cover in this session. We are going to study about the glomerular filtration rate, juxtaglomerular apparatus, followed by the renal autoregulation of GFR, negative feedback control of GFR, sympathetic control of GFR, and peritubular capillaries. Then we will be studying about the DCT and collecting duct, followed by the counter current exchange system. So these are the topics that we are going to cover in this session. So let us see what is meant by GFR, which is the abbreviation of glomerular filtration rate. Your glomerular filtration rate is nothing but the filtrate that is formed per minute in your nephron. Your filtration coefficient, which is nothing but the Kf, depends on permeability and surface area of the filtration barrier. If you see, your glomerular filtration rate is obtained by multiplying your NFP with your filtration coefficient Kf. So which is approximately equal to 125 ml per minute or 180 liter per day. If you see, 99% of the filtrate is reabsorbed and 1 to 2 liter of urine is excreted per day. So this is about your glomerular filtration rate or GFR. So let us see. So we are going to see about the effects of GFR abnormalities. So if your filtration rate is more, then the urine output rises. So if there is an increase in the urine output, it leads to the dehydration of your body and there will be an electrolyte depletion. If your GFR or the filtration rate decreases, then it says that your wastes are reabsorbed into your body. So in this condition, you may be subjected to azotemia. Your GFR can be controlled by adjusting your glomerular blood pressure. So this can be controlled by autoregulation, sympathetic control, hormonal mechanism, which includes the renin and angiotensin. If your GFR is around 60 to 120, then the GFR is normal. If in case your filtration rate is between 15 to 60, then it indicates that your kidney has been affected by some disease. If the rate is between 0 to 15, then it indicates that you have a kidney failure. So this is how your filtration rate determines whether your kidney is functioning properly or not. So now you can see the juxtaglomerular apparatus. In this, we have the podocytes, which you can find it over here, and the sympathetic nerve fiber. So here is your juxtaglomerular cells and mesangial cells. The arteriole over here is the efferent arteriole, and this is your afferent vasomotion arteriole. You can see that your arterioles are covered by smooth muscles, and this is your distal tubule, and this opening is your macule densa. So this gives you your juxtaglomerular apparatus appearance. So this is how it appears. Now we are going to do the renal auto-regulation of GFR. So when you regulate your glomerular filtration rate, which is a GFR, in first case we are going to constrict the afferent arteriole and dilate your efferent arteriole, which is in the sense we are going to constrict, we are going to reduce the flow of the blood in your afferent arteriole. So at that point we are going to dilate the efferent arteriole. In this condition your BP rate increases because we are not letting go the blood in your afferent arteriole. In a second condition, we will be dilating the afferent arteriole and we will be constricting the efferent arteriole. In this case, your BP decreases because the blood that is flowing for filtration is less. So this is how we can regulate the GFR. And to obtain a stable BP range of say 80 to 170 mmHg, which is systolic, we cannot compensate for extreme BP. Next, we are going to study about the negative feedback control of GFR. If you have a high glomerular filtration rate, which in turn says that there is a rapid flow of filtrate in your renal tubules, because if filtration is more, the filtrate will be more. So since your filtrate is more, you can identify that there will be less reabsorption of the fluids, since most of them are let through your filtrate. In this case, your macula densa senses high sodium chloride, that is NaCl concentration in your filtrate, and it will also sense that it has unidentified paracrine secretion. So in this case, we will be constricting the afferent arteriole. When you constrict the afferent arteriole, there will be a reduced GFR, that is the reduced glomerular filtration rate. So this is how we can give negative feedback control of GFR. Next, we are going to see about the sympathetic control of GFR, which is glomerular filtration rate. How to do the sympathetic control is what we are going to study over here. So this is your Bowman's capsule, and inside which you will have the glomerular capillaries. And the one that is on your left is your afferent arteriole, through which the blood flows into your glomerular capillaries. And the one on your right is the efferent arteriole, through which the blood flows out of your glomerular capillaries. So in sympathetic control, we are going to control the filtration rate by blocking or releasing the afferent or the efferent arteriole. If there is a strenuous exercise or acute conditions, that will lead to stimulate the afferent arterioles to constrict. So here you can see that your afferent arteriole is constricted. This will reduce the flow of the blood that is entering into your glomerular capillaries. So once the blood that is entering your glomerular capillaries decreases, the blood that is going out will also decrease, which will result in a reduction of the filtration rate and the filtrate. So when your filtration rate decreases, your urine production also decreases. And in this case, when your urine production decreases, you will be redirecting the blood flow to the heart, brain and skeletal muscles with the most of the fluids. So this condition is depicted in this graph over here. You can see that your filtration rate is decreasing with the increase in your afferent arteriole constriction. So the next case is when you constrict your efferent arteriole, and when you dilate your afferent arteriole, the blood that is flowing into your glomerular capillaries will increase, but whereas there will be a decrease in the blood flow that is going out and will increase the pressure of the blood inside your glomerular capillaries. So which is depicted over here as when your efferent arteriole constriction is increased, there will be an increase in the filtration rate as the blood undergoes a high pressure, and gradually it tends to decrease. Your afferent arterioles and efferent arterioles are a group of blood vessels that supply blood into your nephrons and which take out the blood from nephrons. So afferent supplies and efferent takes out. Next we are going to study about the peritubular capillaries. In peritubular capillaries, the blood has unusually high COP, and your BHP is only around 8 mmHg or lower when constricted by angiotensin II, and this case favors reabsorption. Your water is absorbed by osmosis and it carries other solutes with it. So that is what is done in your peritubular capillaries. So you can see over here your peritubular capillaries which runs around your glomerulus and nephrons. Next we are going to study about the proximal convoluted tubules, which is abbreviated as PCT. Your proximal convoluted tubules are found in your nephron and it reabsorbs about 65% of the glomerular filtrate to your peritubular capillaries. It has a great length and a prominent microvilli, and it has abundant mitochondria for active transport. It reabsorbs greater variety of chemicals than other parts of the nephron. Your cellular route through epithelial cells of PCT, that is your proximal convoluted tubules, and your paracellular route is between the epithelial cells of PCT. So this is how it reabsorbs a greater variety of chemicals, either through the transcellular route, that is through the epithelial cells, or paracellular route between the epithelial cells of PCT. Transport maximum is nothing but when the transport of proteins of plasma membrane are saturated. In this case, the glucose is greater than about 220 Ng per dl, and it remains in the urine, which is glycosuria. So this is about your PCT, which is known as proximal convoluted tubule of the nephron. We are going to study about the tubular secretion of PCT and nephron loop. In your nephron loop, the waste removal will remove the waste such as urea, uric acid, bile salts, ammonia, catecholamines, and many drugs. So these are the wastes that are removed by your nephron. And to balance the secretion of hydrogen and bicarbonate ions in your body, which regulates the pH of the body fluids, is done in your nephron loop, which does the acid-base balance by secreting hydrogen and bicarbonate ions. The primary function of your nephron loop is to conserve the water and also involved in electrolyte reabsorption, which prevents the body from dehydration. Next, we are going to study about the DCT, which is your distal convoluted tubule and collection duct. If you see, your kidney has a gland which is located above it, which is known as your adrenal gland. Your adrenal gland will secrete aldosterone, which controls the urine production. If there is a decrease in your BP, it causes angiotensin II formation. So your angiotensin II will stimulate the adrenal cortex. So when your angiotensin stimulates your adrenal cortex, it secretes aldosterone. So this aldosterone on generation produces and promotes sodium ion reabsorption. So when sodium ion is reabsorbed, it promotes the water reabsorption indirectly. So once your water reabsorption increases, the urine volume that is generated will turn to be low. And this causes your BP to drop less rapidly. So this indirectly says that when your BP drops, the volume of your urine that is secreted will be reduced, and which in turn says that your BP has dropped down. In this condition, the sodium absorption will be high and reabsorption will be high. So this is the effect of aldosterone. Next we are going to study about the effect of atrial natriuretic factor, which is ANF. In atrial natriuretic factor, if your BP increases, that is your blood pressure increases, it stimulates the right atrium, and the atrium will secrete ANF, which is your atrial natriuretic factor. So once the ANF is secreted, it promotes the sodium ion and water excretion. So once your urine volume will increase, and this decreases the BP level. Next we are going to study about the effect of ADH. Dehydration will stimulate your hypothalamus. Your hypothalamus stimulates the posterior pituitary, which is located behind your brain, and the posterior pituitary will release the ADH hormone. If the levels of ADH is increased, it leads to water reabsorption. So once your water is reabsorbed a lot with the increase in ADH, the urine volume that is produced will become down. So this is the effect of ADH production. Next we are going to study about the collecting duct concentrates urine. Osmolarity is 4 times X as high deep in medulla. The medullary portion of the collecting duct, that is CD, is permeable to water but not to sodium chloride, NaCl. So this is how the urine is concentrated in your collecting duct. Next we are going to study about the control of water loss. Producing hypotonic urine, the sodium chloride that is reabsorbed by the cortical collecting duct. So once your sodium chloride is reabsorbed, your water remains in the urine. So it produces hypotonic urine. If the GFR, that is your filtration rate, drops, which increases the tubular reabsorption, so less amount of NaCl remains in your collecting duct. So when the NaCl remains in the collecting duct, it will lead to the increase in ADH, which is your collecting duct's water permeability. Then more water is reabsorbed. When more water is reabsorbed, then the urine is more concentrated. So when it is more concentrated, the urine that is produced is known as hypertonic urine. Next we are going to study about the counter current multiplier. In counter current multiplier, the main purpose is to recapture the NaCl and return it to your renal medulla. If this is your Henle's loop, then the descending limb reabsorbs the water but not the salt, and hence it concentrates the tubular fluid. Whereas your ascending limb will reabsorb the sodium, potassium and chloride ions, that is Na, K and Cl ions. And this maintains a high osmolarity of renal medulla. And this is impermeable to water. Recycling of urine can be done by collecting duct in medulla. The urea accounts for about 40% of high osmolarity of the medulla. So this is about the counter current multiplier. Next we are going to study about the counter current exchange system, and it is formed by vasa recta and it provides a blood supply to the medulla. It does not remove NaCl, that is your sodium chloride, from medulla. The descending capillaries, your water diffuses out of the blood in your descending capillaries, and the sodium chloride diffuses into the blood. Whereas in your ascending capillaries, the water diffuses into the blood and your sodium chloride diffuses out of the blood. So water enters, whereas your NaCl diffuses out. In descending capillaries, water diffuses out and your sodium chloride diffuses into the blood. So this is how the counter current exchange system happens in your capillaries, by exchange of water and sodium chloride ions. Next we are going to see about the summary of the tubular reabsorption and secretion. So this diagram depicts that how the tubular absorption and secretion is done in your nephron. In this region, in your glomerular region, you have the urea, uric acid and creatine and some drugs. And here it will absorb the glucose, amino acids, proteins, vitamins, lactate in urea and uric acid. The water is absorbed in your Henle's loop, and which will generate the urea. Your sodium, potassium and chloride ions are produced over here, which in turn will produce your hydrogen, potassium and ammonia. So finally you'll be getting urea. Thank you so much for joining GTech on glomerular filtration rate. Hope you would have got a very good idea about the glomerular filtration rate.